Hello, biology. The purpose of this video is to give you an opportunity to see the types of questions that you're going to need to know on the Chapter 12 unit test. A great way to prepare for this test is to see the questions, look them up in your study guide, or, write down what you think the answers are, assuming you already know them, in advance of the test. Couple quick reminders for you. You get to retake a maximum of 2 tests per semester. So if you've already taken 2 retakes this semester, you will not be able to retake another one, which obviously would make this test very important to you. If you've only retaken 1 test and you do poorly on this test, you will be able to retake it, and we'll have to work out the schedule for you to do that. Also, because this is a video, I will not be giving you as much time to look up the answers as I do in class. I will warn you and let you know when I will be showing you the answers, so you can pause the video if you are still trying to figure them out on your own. All right, without further ado, here we go. I do want to apologize in advance. I'm drinking some coffee, so you might hear me sipping my coffee or drinking my water, and I'm also grading some papers, so you might hear some shuffling of papers, so I apologize if that distracts you. All right, we're going to do 1 column at a time. Let's start with DNA experimentation and some random questions. Which scientists proved that bacteria are transformed by DNA? Give you about 20 seconds to look this one up. Answers coming in 5, 4, 3, 2, and 1. That was Oswald Avery. Next questions. Which scientist contributed greatly to the discovery of DNA structure but did not win the Nobel Prize? If Hershey and Chase would have found both 32 phosphorus and 35 sulfur in the bacteria within their experiment, what would this have meant? 1 point extra credit. What are the names of the 3 scientists who did win the Nobel Prize for DNA's structure? Give you guys about 45 seconds with this one. All right, answers coming in 5, 4, 3, 2, and 1. Okay, so that was Rosalind Franklin, of course. So remember, phosphorus is tied to DNA. Sulfur is tied to proteins. So that would have meant that if you found them both, the virus's protein coat and DNA were both injected into the bacteria. Of course, that's not what happened. They found only phosphorus, indicating it was DNA that was injected into the bacteria. And Watson, Crick, and Maurice Wilkins also were given the Nobel Peace Prize. When Griffith mixed heat-killed disease-causing bacteria with harmless bacteria, what happened to the mouse? Sketch a molecule of DNA. True or false, genes do specify the instructions for making proteins. Give you guys about 45 seconds with this. Answers coming in 5, 4, 3, 2, and 1. So the mouse died, okay? And that's what told them that something was being transformed. True, genes do specify the instructions for making proteins. Genes code for proteins. And of course, the famous DNA structure is a double helix. What were the names of the 3 scientists who did win the Nobel Prize for the structure of DNA? Hold on a second. I've got that in there twice. That should be easier. True or false, in eukaryotes, DNA replication starts at a single location and occurs in only one direction. Blanks contain instructions for making proteins. Give me 1 second. I'm going to make a quick correction. Sorry. Get rid of this. Problem solved. All right. Showing you the answer in 5, 4, 3, 2, 1. Okay, so that's Watson, Crick, and Wilkins. False. In eukaryotes, DNA replication starts at hundreds of locations and occurs in both directions. Otherwise, it would take forever. And genes contain instructions for making proteins. All right, and give me 1 second. Correction here also. All right. Beg your pardon for the little interruption here. All right, on to DNA structure and random questions. Due to base pairing rules, adenine always bonds with blank and guanine always bonds with blank. Answer coming in 5, 4, 3, 2, and 1. All right, so that's thymine and cytosine. A with T, C with G. T with A, G with C. Erwin Chargaff found that the percentage of blank was about equal to the percentage of blank. This answer is not adenine, thymine, or cytosine and guanine. What are the names that describe those things? What is the name of the shape/structure of DNA? All right, answers come in 5, 4, 3, 2, 1. All right, so they're called purines and pyrimidines. Okay, double helix is the name of the structure we gave DNA. RNA contains the nitrogen-based thymine, DNA contains the nitrogen-based thymine, but RNA has blank instead. Nitrogen bases are connected to one another by blank bonds. DNA is free-floating, meaning no nucleus, in what types of organisms? Sorry about the noise. My oldest son is heading off to work. All right, showing you the answer in 5, 4, 3, 2, and 1. All right, so uracil instead of thymine, weak hydrogen bonds, and prokaryotes, which are the opposite of eukaryotes. Prokaryotes are very primitive. Eukaryotes, like us, are very, very complex. Deoxyribose, a phosphate group, and cytosine make up a blank of DNA. Deoxyribose and the phosphate group alone make up the blank of the DNA molecule. What's the name of the location where DNA replication occurs and blank molecules are made during transcription? Answers coming in 5, 4, 3, 2, and 1. All right, so those three things together make up a nucleotide, sugar, phosphate, backbone. Replication actually takes place at what's called the replication fork, and RNA molecules are made during transcription, specifically messenger RNA molecules. Okay, on to DNA replication. If the original or old strand of DNA being copied and has a base sequence of TGA, GTC, AAC, what will the complementary or new DNA sequence be? So just use your base pairing rules. Also, if you're watching this prior to taking the test, do not mark on my test. Use the back of your Scantron or ask for some scratch paper. Don't mark on my test. Cleared. Thank you very much. I would appreciate that. And of course, if you're ever taking a test and it's been marked on, you have no idea who took that test before you. Do not trust what they have written on it. Answers coming in 5, 4, 3, 2, 1. All right, so T A G C A T G C T A C G A T A T and C G. The double helix unzips, freeing up both strands to be used as templates. The process where DNA is copied, producing two identical molecules, is called what? After this process, each DNA molecule has one blank strand and one blank strand. Answers coming in 5, 4, 3, 2, and 1. All right, so that's called DNA replication, and each new DNA molecule is half old or original, the template that's used, and half new or complementary. So old, original mean the same thing, new, complementary mean the same thing. What's the name of the enzyme that breaks weak hydrogen bonds during replication? In eukaryotes, DNA is found where blank builds the complementary strand and also proofreads for errors, and DNA tightly coiled around histone proteins is called what? All right, 5, 4, 3, 2, 1. Right, so it's the helicase enzyme that breaks those bonds and exposes the two templates. DNA polymerase builds and proofreads, and chromatin is what DNA wrapped around histone proteins is called. How many chromosomes do all human body or somatic cells have within them? What are the missing names if they are listed from small? What are the missing names if they missing names in the blanks if they are listed from smallest to largest? So DNA would be the smallest, chromosome would be the largest. There's a diagram in your book that will help you with this one. All right, answer is coming in 5, 4, 3, 2, and 1. All right, so every cell's got 46 chromosomes. DNA is the smallest. Next smallest is nucleosome, then a coil. You might have guessed supercoil, obviously bigger than a coil, and then supercoils coil even further into the chromosome. So, DNA smallest, chromosome largest. All right, protein synthesis. Nucleic acids are blanks made up of many smaller blanks. You guys, about 30 seconds for this one. Answer is coming in 5, 4, 3, 2, 1. All right, nucleic acids are polymers made up of smaller units called monomers. Each blank molecule has a triplet code called an anticodon. During the process of blank, Amino acids are joined together at the ribosome, and transfer RNA brings certain amino acids based off of messenger RNA's codons. The answer's coming in 5, 4, 3, 2, 1. Each transfer RNA molecule has a triplet code called an anticodon, and during the process of translation, amino acids are joined together at the ribosome, and transfer RNA brings certain amino acids based off of messenger RNA's codons, which are indirectly DNA's code. Blanks are made by building long chains of amino acids, whereas messenger RNA made during transcription. And how many nitrogen bases are needed to specify 15 amino acids? Answer coming in 5, 4, 3, 2, 1. Proteins or polypeptides are made building long chains of amino acids. Messenger RNA is made in the nucleus, and it's 45. So, for every amino acid, you need 3 nitrogen bases. Okay? And remember, 3 nitrogen bases are a codon in messenger RNA. Let's say you have 9 amino acids. That would be 27 bases. Let's say you have 3 amino acids. That's going to be 9 bases. Let's say it's going to be 2 amino acids. That's going to be 6 bases. You will need to know that conversion. So it's a 3-to-1 ratio. 3 nitrogen bases for each amino acid. Okay, oh my goodness, hold on. Something happened to my formatting here. That's not going to work. I'm going to move you over here. Move you back up here. Sorry about that. Hopefully it'll stick. All right, what's it? I, E, G, H, and J. Here's I. Here is E. Here is G, H, J. All right, answer's coming in 5, 4, 3, 2, and 1. Okay, so this growing thing, is a polypeptide chain. This is an amino acid. So these are all amino acids that are being bonded together with peptide bonds. So this is an amino acid here. G is the anticodon. So these 3 nitrogen bases are the anticodon because this is transfer RNA. Of course, H, these 3 are the codon. These are being read at the ribosome. All right, so hopefully those are the hardest ones. You're going to have to label all these other structures as well, but those are the ones that cause the greatest confusion, and it's matching really, so you should be fine. On the mutations and random questions, what kind of mutation is never a frame shift mutation? Answers coming in 5, 4, 3, 2, and 1. Substitution mutations are never frame shift. Now, they may actually cause damage to a protein, damage to the organism, but they are not going to shift the reading frame. Remember, the reading frame is the 3 bases on messenger RNA are read at a time, because those 3 bases in the codon coincide with the anticodon, which coincide to a specific amino acid. What category of mutation is an inversion mutation? RNA polymerase is responsible for making blanks in the cell's nucleus. All right, answers coming in 5, 4, 3, 2, and 1. All right, so those are chromosomal. So inversion, deletion, insertion, and translocation are the 4 types of chromosomal mutations. Remember, the other type of mutation is gene mutations. Okay, RNA polymerase makes messenger RNA in the cell's nucleus. All right, so given DNA strand 1 and DNA strand 2, you'll notice when DNA replication is taking place that this strand is different than this strand. What type of gene mutation is that an example of? Mutations are always blanks to genetic material being copied. Insertion, deletion, and frame shift mutations could change every blank, causing the protein to potentially be non-functional. All right, answers coming in 5, 4, 3, 2, and 1. All right, so that's a deletion. You can see that there was a base that is omitted there. Mutations are changes in the genetic material or changes to DNA, and a frame shift mutation could be very damaging because every single 3-letter code after that mutation could impact every single amino acid. And of course, that's very likely to make the protein non-functional. Last questions. What are 3 things found in both DNA and RNA? RNA, tRNA, and mRNA are all involved in what? All right, answer coming in 5, 4, 3, 2, 1. Okay, phosphate groups and either adenine, cytosine, and guanine are found in both. Remember, ribose and deoxyribose are different. Thymines only in DNA, uracils only in RNA. And all 3 types of RNA are involved in protein synthesis. All right, you guys, I hope you found this video helpful, and good luck on your test.